Okay, so as I said, we're going to be talking about the nervous system. So here we have an individual who basically his arm has been amputated, and what do you guys think? So in terms of robotics, so this guy actually has a robotic arm, and it's being controlled partially by his actions and what he does. So do you guys think at some point we're going to be able to fully restore, say, appendages that have been amputated or removed?
Restore in what manner? Do you mean
In terms
like
of, I mean,
with
currently
all of our
we have
organic
them.
flesh and all, or you know what I mean, organically, or you mean restore its function robotic?
Well, robotically, we have the function partially restored. It's not full function, but partially.
Yeah.
So if we take a look at this individual, if you take a look here at his arm, right here. Oh, okay. Let me switch this. Okay. So take a look at this individual's arm here, right? So he actually has an artificial limb there already on that arm. And so it's a robotic arm itself. And so it has some of the capability, right? But not full capability. So what is it that is currently limiting our ability to get that full capability?
Or probably our synapses, like they can't get the actual robotic arm to respond to commands from our nervous system as efficiently as our limbs, our natural limbs do.
Okay, so exactly. So the fact is that once those nerve endings are kind of damaged, and those pulses, the impulses, we're not able to translate those signals from those impulses from those nerve endings. One, because they've been damaged. Secondly, because that translation isn't available yet. We're getting there. We're getting pretty close. As you can tell by this individual is able to somewhat grab the functioning, the foosball handle, right? So today we're going to kind of talk about that nervous system and kind of the makeup of it. Generally, we're not going to talk about the anatomy, but more the physiological aspects of it, okay? Okay, so we have the central and the peripheral nervous system. So the peripheral nervous system is basically going to be made up of ganglia or ganglion and nerves. And you can kind of see the structures here on our little diagram. And then our central nervous system is going to have similar makeups, but they're not going to be as obvious as what we see, and they're not as easily dissectable and stuff like that, because in that context, we're looking at the brain and the spinal cord, things like that. So peripheral is everything else other than the brain, okay? So if we look at the brain is made up of gray matter and white matter, okay? Let's see. The gray matter basically makes up the outer cortex of the brain. And it actually surrounds the white matter of the brain, okay? Okay, so when we Okay, so the gray matter is basically going to be a lot of cell bodies and dendrites. Let's see. This is going to be where your axons are. So the cell bodies and the dendrites are going to act like a protective coat for your axons to kind of keep those guys messed up. Okay, and you guys remember that your axons, they're going to be coated with the myelin. Okay, and so that's just going to be kind of like that insulation. And it's myelins are going to be kind of it's a lipid-rich protein that coats your axons itself to make sure that your signals are being transmitted and not being lost along. Okay, so in chemistry, the nucleus is where the protons and neutrons are at, kind of the center of the cell, and then you have the electrons surrounding it. In a cell, the nucleus is where all the DNA is contained, okay? And it kind of controls all the aspects of what's going to be happening in the cell itself. Kind of like the protons kind of define what the atom is, okay? In the case of an atom, okay? Now, the central nervous system is so I'm sorry, the nucleus in the central nervous system kind of controls all aspects of what's going to be happening in the periphery as well. Okay, so very similar, that term nucleus is being displayed here. And so this is basically the nucleus of the brain. And it's going to be basically the central function of the cell bodies of neurons. And you can kind of see that circled in the red right there. Okay. Okay, so we have the optical nerve versus the optical tract. Okay. So that's basically start off with the definition. So these guys are just going to be bundle of axons. And these guys are going to be found in the CNS, where nerves are the same thing, bundles of axons. Say fibers found in the periphery, PNS. Okay. So if we take a look here, the optical. So it's located in the central nervous system, where the optical tract is located in the central nervous system. So it's actually located in the inner part of the brain, where the optical nerve is located at the periphery on the outside, closer towards the eye. Okay. Questions, concerns, Cash? In your book, there's a nice little table. It says, so your cell bodies, so neural, cell bodies. And CNS, they're called the nucleus. In the PNS, they're ganglia or ganglion. Okay. Okay, so what are some of the basic functions of our nervous system?
Basic functions of what?
Of our nervous system.
The nervous system is responsible for our movements, commands
Okay.
voluntary movements.
You said involuntary movements.
Voluntary.
Voluntary.
Yeah, voluntary.
Okay.
And involuntary. So the autonomic nervous system.
Okay. Okay, so when you say it's responsible for our movements, why? How and why? Well, I kind of know how, but
So are you asking how, or are you asking why?
I'm asking, let's start with how.
Our brain sends signals through our nerves into our limbs, etc.
And why does our brain send signals to our nerves? What is the reason for the significance of that?
To notify it if it needs to move or if it needs to to move, and like let's say you touch a stove, to move right away.
So in order to respond.
Yeah.
Okay. So our brain is sending signals for us to respond. So that causes us to move or to do something. Our hand off the stove, or right. So then we're responsible for response. But how does it know that it needs to respond?
Through stimulus.
Okay. So it gets some type of stimulus that kind of tells it, hey, you need to respond. So it needs to be able to sense stuff and then also respond. Break it down simple, right? Mm. Okay. So if we think about function is kind of broken up into those terms, right? Sensing, sensations, and then response. Okay. And so if we think about our brain itself, it's going to be kind of set up in that format. You're going to have regions of your brain that's going to be responsible for sensing, and then other parts of it that's going to be responsible for responding. Okay. So, and that kind of brings us on those senses. What are the senses? What do we sense?
Pain.
Pain. Ooh, yeah. So touch, right? Pain.
Temperature.
Okay, so we can sense temperature. Okay. So if we do this kind of more generally, right? Because temperature, you know, it could be temperature in terms of touch. It could be temperature for, I guess, the air itself. So what are the 5 senses?
Touch. Taste.
Smell.
Smell. Hear.
Hear. See.
Yep. Yep. So I guess it's like hear no evil, see no evil, taste no evil, right? Smell no evil. Okay. So those are all of the senses, right? And all of those guys are going to be stimulated by, as you said, touch is going to be a physical aspect. It's going to be mechanical, right? Sight is going to be a stimulation via light. So those wavelengths of light. Hearing, you know, is going to be sound, and you know, smell has that chemical aspect of it, right? Because odor is a result of a chemical, and taste also chemical, right? Something that's very bitter, like my coffee this morning, right? Okay. So we have sensations, we have response, and then we have integration. Integration is where we basically we combine both aspect of sensory with memory or learning. Okay, and so those are going to be kind of like the 3 major functions, you know, that response, the integration, and sensory. I'm sorry, response, sensation, and
So does response describe something that's more reflexive, like when we touch something that's hot?
yes.
Like,
So it's more like the motor functions. Yeah.
And so then something like integration is more like muscle memory, when you like say remember like
Yes.
like a combo when you're boxing, and then that just becomes like sort of reflexive over time.
Yes. Yes. It is. It is just like that. Yeah. That's a good analogy. I have to use that one. Okay. Come on. Sorry. Give me a second while I figure out where my, okay, there we go. Okay, so this is the breakup of the somatic, the autonomic, and the enteric structures of the nervous system. So the somatic structures are going to include our spinal nerve, and so that's going to be our motor fibers and our sensory fibers. And it's also going to include sensory ganglia. The autonomic structures. So that's going to be the sympathetic and the parasympathetic ganglia. And then the enteric nervous system is going to include nervous tissue around the digestive organs. So these guys are somatic. Actually, it's a combination of both somatic and somatic and autonomic here. So ganglia is going to be a combination of both. And then we know that this guy's going to be somatic and autonomic, and this guy's going to have combinations of both as well. Okay. Okay, so this is a functional magnetic resonance imaging. And so basically, this lets us know how active your brain is going to be. So in this case, this is actually using visual stimulus, right? And so they're probably shining lights in somebody's eyes or something like that. And as a result of it, they notice that in the back of your head, you're actually you're getting a lot more signals towards there, okay? And so that just kind of confirms to where we talked about the optical nerve, and then in terms of the central nervous system, we have the optical tract. And so that's the location where the optical tract is at. And so it just kind of confirms that result, right? Come on, sorry. Okay, so let's talk about the neuron. So the basic structure of the neuron is shown here. So we have the axon, which is basically that length where you're transmitting the electrical impulse. We have the cell body. Of course, you see the cell membranes, and we have our dendrites, which are going to be the ones that are going to be extending. And extending out to the other cells for communication. And then you also have down here your synapsis, where you're actually secreting or the signals that are going to be transmitted. So you can see the myelin sheath, which serves as a protection. We have our oligo dendrocyte, which basically makes those myelin sheaths. Okay. So neurons are basically classified by their shape. They're unipolar, basically meaning that you have the cell and its nucleus located in one part, and then unipolar meaning that it's extended away. The cell and nucleus are below it. So it's bipolar, where the central part of the cell is located in the center, and then the dendrites are extended both ways. And then we have multipolar, and that's kind of like going in all directions, but you have the axon going out of it. And so you can end up with the cell body itself with multiple dendrites coming off of it. Okay. Simple, easy to distinguish between them. So there are other classifications of neurons. So we have what are called pyramidal cells. And so these guys are located in the cerebral cortex. Excuse me. And I'm going to butcher this so bad. Putative cells, which are these guys are going to be located in the cerebellar cortex. And we have our olfactory cells. And so these guys are going to be located in the olfactory epithelial and the olfactory bulbs. Okay, and okay, so this is the ganglia cells of the CNS. So the CNS basically has astrocytes, they have oligodendrocytes, they have microglia, they have appendium cells. And so these guys serve as a support. The astrocytes. We know that these guys are responsible for insulation. So these guys are going to be involved in the immune surveillance. Okay, I apologize if I misspell things. I am not an English major. So and then under certain circumstances, since they're surveilling, they'll be taking samples. So in other words, anything that's coming in there that doesn't seem normal, they'll engulf it, they'll chew it up, and then they'll actually give signals for the for your immune cells that start letting you know what's going on there. Okay. See, these guys, where are they? Up right here. So these guys are going to be creating your CSF.
What is CSF again?
It's your central spinal fluid, I believe. Your cerebral spinal fluid. Let's see. Okay.
So
And
our
what was that?
spinal fluid, does that act like a conductor?
It's basic. Well, okay. So basically, what it is, it's blood that's kind of been filtered, and it basically is just circulating in your brain or in your central nervous system. Okay, so because you have your blood-brain barrier, right? So basically, your blood-brain barrier basically kind of allows for the filtration of that blood. These cells are responsible kind of for that filtration, as well as putting other things in, right? Other things that that fluid. Okay. And so they filter the blood at the blood-brain barrier. And then that stuff that's filtered through is what's able to go in there. And so during that process of doing that filtration, they also put in some extras, some other proteins and stuff like that, that are necessary for things that are in. Okay, so I guess I should probably elaborate a little bit. So you guys have a blood-brain barrier, and so that kind of, so your blood is very, your brain is very sensitive, right? And if certain things get through there, it can cause havoc. And so there's a nice little barrier that kind of keeps all of those, like, let's say, broken up cells or large particles, things like that, keeps those things from coming in there because they can kind of gum up that system and clog it up so it doesn't function the way it does. It doesn't impulses the way it should. And so that's basically, it's like a protective barrier to kind of keep all damaged stuff out, and any large particles or very large chemicals and stuff out of there, so they don't affect what's going on in your brain.
So it's basically like a membrane around the.
Yeah, but it's a little more than just the membrane. It's not like an individual membrane. It's those cells, particularly these cells, kind of serve as that line of defense to kind of do that filtration. Okay, so but there's only certain things that are able to come through there, right? So those are very, very small particles. So those are like nutrients, like sugar, because we need a lot of sugar to keep our brain going. They're amino acids. They're, but then there are other things that can't, like larger molecules that are unable to go there, like long lipid structures, heavy lipid structures. They're not able to sit there and make it through. So it does a really good job of selecting what can go in, and primarily things that are very small are able to get through, where things that are larger aren't. Does that make sense?
Yep.
Okay.
So if
Yep.
if the CSF is not working well, can it cause some headaches?
Yes. Let's say, for instance, if you've been eating a lot of salt or something like that for a while, yeah, it can cause headaches. Your body is salt, and you say you're getting salt that's going across that barrier, it can cause issues. It depolarizes your whole nervous system down there and causes other things to go on. So like high blood pressure, things like that. Okay, let's see. Okay, so your PNet, so these are, come on. These are, oops. I forget something. Okay, can you guys, yeah. Sorry, my computer's giving me a little bit of trouble today. Okay, so these are this is the ganglia cells of the PNS. Okay, so the Schwann cells, they're going to act just like the oligodendrocytes. So they're going to make an insulation around your axons in the PNS. So they make that insulation. Your satellite cells are going to be there for support, And so they're just like your astrocytes in the CNS. Okay. Okay. And then this insulation, again, these are going to be your, still, it's going to be myelin, but they're just being produced by the Schwann cells. Okay. There we go on time. Okay, so we have, so this is basically the process of myelination. Myelination is basically wrapping the axons with the myelin, okay? Okay, so in this case, you have the cell itself is actually wrapping itself around the axon. So then it's actually going to be secreting the myelin to form that myelin sheath. So as it continues to wrap itself around. Okay, so Schwann cells are going to be able to do this in the peripheral nervous system, and then the oligodendrites are going to be doing this in the central nervous system. Okay, and this is just basically an image showing you the axon being wrapped with the myelin sheath. Okay, so let's talk a little bit about that function. So remember we were sort of talking about like different parts of your brains are going to be responsible for different things. And so it all depends where everything is going to be wired. Okay, so if you're like when we jump in the shower, we're actually testing the water, right? You guys don't jump in immediately because we want to make sure it's either not too hot or not too cold. Right? So we have, you know, in our body, in our skin, we have what we call thermoreceptors. And these thermoreceptors are going to sit there and recognize whether something, the temperature is too hot or too cold. Okay. So the first thing that we're going to do is if we jump in the shower, we touch the water, right? So we're going to get that sensation. So that touch is going to sit there and let us know. It's going to send a signal. So we're going to have an action potential that is going to be going along the axons. Okay. It's going to go up here to our sensory axon, where it's going to enter the spinal cord and then the synapses of the brain. So then that potential goes over. So this is the spinal cord and the synapses where they meet. And so it's going to send that signal across. So this is the inner neuron. It's going to actually pick up the signal, and that signal is going to continue going until we get to the left side of the brain. So the left side of the brain is going to be where we have, we'll be able to sit there and process that response. Okay, the water's hot, or hey, the water's cold. Move your hand, you know. So then as a result of that, right? So, and this is going to be at the cerebral cortex. So then we process it, we get a response, and then we say, okay, we need to move something, right? Move your hand, or, you know. So then it starts to send that potential, that action potential down this path. And then we get to the spinal cord. Spinal cord, we have here the upper motor neuron contacts the lower motor neuron, continues to send the signal, and then we respond, right? So we get down here, the lower motor neuron causes us to sit there and move, contract, based upon, oh, it was too cold, let's move, right? And then we adjust our temperature, okay? Questions, concerns, Cash? Okay. So, closer look. So we first have that sensory input, right? We touch the water's hot or the water's cold. Either way, if it's hot, we're gonna move. If it's cold, we're gonna move, right? It's a natural response. But we're gonna respond as a result of that. So if we first get that input then sends a signal to our nervous system. And then we have the response, that motor response, move your hand. Okay. Make sense?
Mhm.
Okay. So next, we're going to be talking about that action potential. Right? So what makes up that action potential? Okay, so this here represents the cell membrane. And you guys remember from the very first couple of chapters, we were talking about the composition of the cell membrane. It has that phospholipid bilayer. You guys can see it nicely here. I wish they'd shown this earlier. So the phospholipid bilayer is composed of phospholipids, and they're connecting to each other, right? So we have that polar head, meaning that it's, or we'll say, hydrophilic head, meaning it likes water. And then we have the hydrophobic tail. Hydrophobic tails are input towards each other. The hydrophilic head is next to solutions or water, so they're able to interact with water. Now, these guys will keep out the water because the membrane itself, these chains are hydrophobic. They don't like water. So they're going to keep water out of that. Other things are going to be able to go through it, but water is not one of those things. Okay. So we have these pores that allows things to go through, such as water or such as nutrients, ions, things that are polar or charged are going to be able to go through the pores. They're not going to be able to go through the membrane. Okay. So this is the outside of the cell. So that's your extracellular fluid. This is the inside of the cell. So that's your cytosol. Okay. These guys are called transmembrane proteins. They're called transmembrane proteins because they go across the membrane. Okay, so this here is a ligand gated channel. So basically, when the ligand binds, the channel opens. So it's kind of controlled by whether or not you're getting binding. So we have acetylcholine. Acetylcholine, we kind of talked about this last week. Its response was one of those first responses. It's a neurotransmitter. It binds to the surface of this gated channel. So that transmembrane actually acts as a receptor. It recognizes the acetylcholine is there, then the channel opens up. When the channel opens up, sodium is able to come in, right? Or potassium is able to go out, or calcium is able to go in. So depending on where we're at and what we're doing, so you notice that we had calcium influx when we were talking about muscles, right? So in that case, calcium was able to go into the cell. Okay. And so, and it's going to flow based upon a concentration gradient. You have more calcium on the outside of the cell. And so as soon as that channel opens up, it diffuses in because a higher concentration on the outside, so it's able to go inside of the cell. Okay, and then that's just the opposite in the case of the potassium. Potassium is going to be going out because concentration of potassium is higher on the inside, and so it's going to be going that way. Okay, so you're able to sit there and control the diffusion of those ions in and out by controlling or by having the presence of your neurotransmitter, your acetylcholine. Okay. So these guys are these are mechanically gated channels. Okay, so in the case of mechanically gated channels, you no longer have the receptor there. So these guys are going to be kind of controlled by pressure or touch, right? So because they're mechanical, right? So if you have some pressure on it, then the gate is going to open, and then you're going to get the flow of your ions in that way. So
In the form of what, though?
what was that?
Pressure in the form of what, though?
As in, let's say you touch something that is hot, right? So you're in that, well, pressure, like, okay, so your finger touches something. So by that physical connection, by touching, I'm putting pressure on that receptor or on that channel. That channel is going to open up as a result of that.
So actual physical pressure.
That's right. It's a mechanical process. Yes. So some type of physical
that's
pressure.
a result of our interaction with our environment.
That's right.
Yeah. I thought it happened at a chemical level inside of us, inside of our bodies.
Yeah. Not necessarily due to a chemical response. It could be pressure, as in pressure buildup as a result of it. That's another process. So you're having a chemical reaction that's your body's producing acid, and then you're producing, you know, your that acid is say eating up something, and you're producing gas as a result of that. Gas as a result of that acidic reaction. So then that causes that. And then, but that's also physical pressure, because you're making, even though you're making it chemically, it is adding pressure to the system because you're making more gas. So it could be along those lines too, as well. It could be a result of it, but the response of this is just done mechanically. So either it's pressure as a result of touching, or pressure as a result of pressure, the physical aspect. Does that make sense?
Yeah.
Okay. Okay, so this guy's going to open up physically, the mechanical. So the same thing is going to kind of happen with our thermoreceptors, right? So it's going to recognize temperatures. The temperature is hot, we're going to open up. If the temperature is cold, it's going to close down. Okay. So we also have voltage gate channels. So these guys are going to basically be they're going to be able to sense charge. So there's certain amino acids, by you having a charge there, these amino acids are going to behave slightly differently. So because these amino acids behave slightly differently, it's going to allow that channel to either open up or close, because it's going to be able to recognize that charge as a result of something positive being there or something negative being there. Okay. We also have what we call leakage channels. These guys are just essentially open, right? And they can, they're open, let's say the leakage channel, when the leakage channel is closed, it doesn't allow the ions to go through. because it moves the ions based upon their electrical properties, whether they're positive or negative. As soon as it opens up, it allows that ion to pass through the membrane. Okay. So questions, concerns, Cash? Okay, so when we talk about potential or action potential, or what we call the membrane potential. So the membrane potential, is just basically charges along the membrane. If we take a look at this guy, the membrane potential is just charges along. So here we have on the cytosol, they're all negative. Your negative charges are distributed along the surface. And then in the extracellular area, so inside of the cell, negative, and then outside of the cell, positive. And so if we measured, if we measured the voltage across the membrane, so in other words, we stick an electrode through the membrane structure, and then we have a reference electrode on the outside, we can sit there and get the voltage across the membrane. Okay. And of course, MV stands for millivolts. That's typically what it's going to be measured in. Now, if we had an equal distribution of it, we're going to have between the ions on the outside and the ions on the inside, then it's going to have a zero charge. If there's an unequal distribution of it, right? So here's our reference, and it's unevenly distributed, then it's going to have a negative charge because we're in the negative region, okay, compared to that positive region. So if we look at the action potential, this is where we're starting out at negative 70. Something happens, we get a response, we get depolarization, right? So that means that those channels are open to allow that plus to go through, so then we can get closer to that neutral charge, right? And then channel close, we'll get repolarization. The first thing is that we'll overcompensate, and then we'll level off. So that's called hyperpolarization. Okay. So again, in terms of the response. So this is where we're at rest. We get a stimulus, channel opens up. So our voltage is going to go higher. Voltage is then going to fall. We go below, and then we're back at rest. Okay. So first step is this polarization or depolarization?
Polarization.
Are you sure? So initially, we're polarized, right? And then we become depolarized because we're going to neutral. Polarized basically meaning we have plus on one side, we have minus on the other side. So if we're opening, let's go back to this one. Okay, so pluses and minuses on different sides of the membrane. This guy is polarized. Polarized basically meaning you have pluses on one side, minuses on the other. Okay, so we get a response, channels open up, what happens? Let's say we had a channel here, and this channel just opened up. What's going to happen to these pluses?
They start to move.
Where are they going to move? They're going to move inside.
Yeah.
So our pluses are going to move inside. And then our minuses are going to start moving outside. Okay, so then you're going to get a point where you're going to get to equilibrium, and in this case, over equilibrium, because what's going to happen is that your pluses are going to be pushing out there to try to get to equilibrium, right? To get balance so that the pluses on the inside equal the pluses on the outside. And then, so the minuses on the inside equal the minuses on the outside. And you're going to get to that point where you have a zero charge or a neutral, okay? And so that's what you're seeing there in terms of that polarization, depolarization. Depolarization when it becomes so that you have balancing on both sides of that plus and minuses. So it's depolarized. So the first step, you get your stimulus, you depolarize, right? Then after your stimulus has been gone, it starts to repolarize because it goes back because it's just a potential. It's just going to happen, and it's going to happen like a cascade along those lines. Depolarize in that area, then repolarize. Okay. So your voltage is going to fall, and then you're going to be back at rest. And so that's the end of your action potential. A graded potential. Okay, so we have a small stimulus. It causes that first, the depolarization, and then it repolarizes, right? Then we have a larger stimulus. It causes more depolarization. So this is the case of when we have a longer stimulus. So we get the length, the width of that polarization is going to increase. Okay, then this case, this guy has a larger stimulus, and it's actually going to be above the threshold to kind of trigger an action potential in the postsynaptic nerve. So we've gone above the threshold. So these guys never reach the threshold. This one does. So this is going to trigger that action potential. And so these are stimuli that result in hyperpolarization. So these are going to kind of be dependent on specific ions, channels that are going to be activated. And so hyperpolarization basically is going to cause more charge distribution between the positive end and the negative end.
So then how does this all translate as far as like in the real world?
Okay, so when we start, we have the stimulus. We touch a hot plate, right?
Mm-hmm.
So if the stimulus isn't great enough, right? If it doesn't reach that threshold, we're not going to move our hand because we're not going to get a response. Okay, so this basically, the stimulus causes that the initiation of this, the action potential, right? But if it doesn't reach above the threshold, we're not going to do anything. This one, in this case, does. Here's our threshold. It goes above that. So then it's going to trigger the a response action potential. So in that case, we get it. It sends the signal. It triggers the response, and then I move my hand. Okay, so this guy is here. We've triggered the stimulation. We've gotten high enough to get a response, and as a result of that, this is going to be me moving my hand. Okay, where when we talk about the gradiated, here, small response. So it's going to depolarize. I'm going to keep my hand there. It's not enough to sit there and cause me to do anything. This one is enough to cause me to do something. So I'm going to have an action potential. Okay, so it's going to continue down the line, send that signal, and then it's going to result in me moving my hand.
And then hyperpolarization, is that when we're going, when our body's not able to maintain homeostasis, like for like
something knocks us off, right? So in the case of hyperpolarization, is when you something's kind of knocked off. So you get some stimulus that kind of knocks you off of what you normally do. So your normal response. So this guy, in this case, you're not gonna trigger a response. You're not gonna trigger a response to it. In the case of hyperpolarization, you're gonna cause it not to respond at all, right? So you've done something to kind of throw it off. So some stimulus causes it to go off, so it's going to be neglected entirely, right? So I'm trying to think of something that, how that would work.
So is that in the case of like damaged nerve cells that aren't responding?
Well, see, it could be a case of damaged nerve cells, but in most cases, damaged nerve cells aren't responding because you no longer have that connection. In this case, you still have the connection. You have the potential to do a response, but there's something impeding it. Like in the case of certain drugs, okay? Certain drugs will cause ion channels to open up. Like, let's see, do you get heartburn? Anybody get heartburn?
Sometimes.
Okay. So heartburn is a result of, well, a result of your cells responding to certain things, right? So obesity, things like that. So as a result, you start making more and more of these receptors on the surface, right? And they're proton pumps, I believe, of your stomach. And so you start making, as a result of those proton pumps, at H+, you're making more and more acids and in your stomach. And so you get like acid reflux. And heartburn is a result of acid reflux. You're producing too much acid in your stomach, and so you're making all of this acid reflux, right? So one way of treating that is to use drugs, right? So there are drugs that inhibit proton pumps. And so I believe it's Prolisac. So it's an over-counter proton pump inhibitor. You take it, it causes you to produce less proton pumps at the surface, or it actually directly inhibits the proton pump. So you're not producing as much acids because you have this drug that keeps those protons from producing protons, which causes your stomach to be more and more acidic. Okay, so that is a specific pump. In that case, it's acting kind of similar to this because protons is plus charge. So that's your H+. So those guys are being kept in cells and not being secreted in the cytosol. So you have that plus being kept in the cells and not being released. Okay, so then that's going to change that function. So in that case, you're going to have that hyperpolarization because you have a certain level of your protons that are there, and they're being pushed down. Do you understand?
Yeah.
So that would be a case of that. But I'm trying to think of in nature, I have to think about that stimulus that we call hyperpolarization naturally without a drug. Okay.
Well, since it's all heavily dependent on calcium ions and all that stuff. So like minerals and stuff that we get in our diets. I mean, is it safe to say that if we're deficient in
Yes.
a certain thing, that it would cause the same thing?
Yes. Yes. If you are deficient, it is going to cause the same thing because your body is trying to compensate for the lack of not having enough. Okay. So here, this is basically postsynaptic potential, and it's summation. So basically, it's a summation of everything that's going on along the entire axis, right? So at point A, okay, so here this is our starting point. Oh, you guys can't see what Okay, can you see it now? Okay, so this is that's me at rest, right? Okay, so we get our first stimulus. Okay, it goes up to a certain point. And then we get another stimulus that kind of pushes us up over a little bit more. So as a result of that, we get depolarization, right? We start depolarizing. Okay, so we start that process of depolarizing. And then it says at point A, we have several different excitatory postsynaptic potentials. And these guys are going to basically add up to a large depolarization. Okay, so this one adds up to get this one, right? And so it's just summing on itself. So we end up with a larger depolarization. At point B, we have a mixture of excitation and inhibition. Okay, so continues adding up. We get a longer depolarization event. We go up here, we get excitation and we get inhibition. And so as a result of that inhibition, we come back down. Okay, so excitation, inhibition. This is us trying to level off. Excitation, inhibition. Excitation, we finally get to the point, we level off. Okay. So in other words, if we have a lot of responses, if we're getting excited by one thing and we get more excitation, you know, I touch something hot, my arm gets hotter, right? I'm in the middle of a fire, right? So excitation, and then I get to the point where this part is getting damaged, inhibition, levels off. It gets excited again, excitation, inhibition. Okay. Okay, so during the inhibition parts, that's where you're going to get that hyperpolarization. So the pink parts, it's going to drop down. So you're leading towards that hyperpolarization. Okay. Questions, concerns, Cash? Okay, so we have synapsis. Okay, so there's going to be the electrical synapsis, and there's also going to be a chemical synapsis. So chemical synapsis is basically a chemical signal. So those guys are going to be like your neurotransmitter. Okay, so we basically send off our neurotransmitters. So those signals are going to sit there and cause our electrical synapsis to occur, right? And so that's going to be that movement of your ions through the channels. Okay. So the thing about neurotransmitters is that one of the benefits, and it can also be one of the drawbacks to neurotransmitters, is that those guys are constantly, they're being made, like they're proteins, they're being broken down, they're being degraded, they're being taken up, you know, and then occasionally they're recycled, but it's not that often. So neurotransmitters, they're going to be binding to the receptor, they're going to be internalized, they're going to be broken down, okay? Or they're going to be broken down in the system if they're there too long, okay? So if you're continuously making them, then they're going to be broken down, okay? So there are different types of receptors. We've kind of touched on it a little bit. we have the ionotropic receptors. So those guys are going to be, they're going to basically recognize ions. And so in the case of your ionotropic receptors, your neurotransmitter is going to bind, and your ion is going to be able to sit there and go through. You're going to have metabolic receptors. So in this case, your neurotransmitter is actually going to bind. It's going to end up causing a signaling cascade. This guy is going to send a signal to this G protein. This G protein is going to activate another protein, an effector protein. That effector protein is going to send another messenger signal, and then that signal is going to cause this guy to open up. Okay. Ooh. Questions? And when we talk about these receptors, so we have receptors that are going to recognize certain neurotransmitters. They're going to be neurotransmitter specific. So only certain neurotransmitters are going to be able to bind to that. So a lot of the drugs that we use to sit there kind of control things, like I said, I was talking to you about the ion pump, the inhibitor of the ion, the inhibitor of the calcium channels, right? So those guys are binding. They're either binding competitively with those neurotransmitters, or they're binding allosterically, basically meaning they're binding to a point so that neurotransmitter can't bind anymore. Okay, and so if that neurotransmitter doesn't bind, then you're not going to get that opening up. Does that make sense?
Yeah.
Okay. Okay. What else should I tell you? So you guys understand that neurotransmitters are proteins. So they'll call them neuropeptides. Okay. I have nothing else for you. We have lab at 11. And so I'll meet you guys in lab.